Hi everyone, my name is Leo Zornberg, and today I'm going to be talking about epitaxial growth of DNA assembled nanoparticle superlattices as part of my 344 term project. Let's start with a quick overview. The epitaxial growth of DNA assembled nanoparticle superlattices is very similar to atomic layer deposition, or ALD. The main difference is that in this system, we're depositing monolayers of nanoparticles rather than monolayers of atoms onto a surface. For this presentation, I will be speaking about the specific system of nanoparticles composed of a gold core that are grafted with DNA, and this DNA is what allows them to form selective bonds. So here is a quick schematic. We see that we have our gold core. Attached to our gold core is an anchor strand that's attached to the gold surface through a thiol bond. The anchor strand of DNA is hybridized to a linker strand of DNA, and the linker strand of DNA is also hybridized to a spacer strand that provides rigidity to the DNA complex. At the very end of our linker strand, we have a sticky end. This sticky end is what allows us to form bonds with other nanoparticles by hybridizing to a sticky end on the other nanoparticle. Now, in this schematic, we've shown one DNA complex connecting the two nanoparticles together. But in reality, the nanoparticles are coated with DNA, so the bonds between them is composed of many different DNA complexes. In order to form a monolayer of nanoparticles on the surface needs to be functionalized with DNA. To accomplish this, gold posts are fabricated on a substrate, and these gold posts are then functionalized with A-type DNA. These gold posts then behave like a perfect monolayer of A-type nanoparticles. Growth off the substrate then proceeds by alternating deposition of B and A-type nanoparticles. The A particles serve as a template during the deposition of the B nanoparticles, and vice versa. After each deposition, the layers are annealed in order to reach equilibrium. In this case, the equilibrium configuration is a cesium chloride structure. Now, let's investigate the process model. We want to describe nanoparticles as they adsorb and desorb from surface sites. If we approximate the nanoparticles as non-interacting, we may use the Langmuir adsorption model, which gives the following relation. The fraction of filled surface sites is related to the maximum surface coverage, the concentration of nanoparticles in solution during the deposition, the adsorption rate constant, and time. Furthermore, the maximum surface coverage is given by the following relation, which is a function of concentration and the equilibrium constant. Both the adsorption rate constant and the equilibrium constant are approximated as Arrhenius, meaning they follow the following exponential relation to temperature. We see that even if the reaction is allowed to run to completion, the surface coverage remains less than 1. This results in a roughening of the surface after multiple deposition steps, since the growth at each specific site is a stochastic process rather than a uniform process. Specifically, the growth at a specific site is a Markov process. The deposition of the current layer is dependent on the previous depositions. If a specific site has gone an even number of cycles without growth, the probability of growth is theta max, the surface coverage. This corresponds to a particle attempting to bind to a complementary surface site. However, if this site has gone an odd number of cycles without growth, then the probability of growth in this cycle is 0, since it corresponds to a particle attempting to bind to a non-complementary surface site. We may use combinatorics to calculate the probability that a single surface site has grown m times after n deposition steps. This probability distribution has the following property. The spread of the distribution will decrease with smaller n and will increase with larger surface coverage. Larger surface coverage corresponds to a larger concentration during deposition, or a smaller temperature during deposition. This roughness may be addressed by an annealing step. This annealing step allows for surface diffusion that will smooth the surface. We may now investigate the process parameters of this deposition technique. Because epitaxial growth of DNA-assembled nanoparticle superlattices behaves very similar to ALD, it has many atomic analogs. Increasing the concentration of nanoparticles during deposition will increase the deposition rate, will increase the final coverage at each step, and will decrease the surface roughness of the final film. Increasing the deposition temperature will increase the deposition rate, decrease the final coverage, and increase the surface roughness. We may also control the activation energy. In this system, the activation energy is largely dictated by the Coulombic repulsion between nanoparticles. This may be controlled through a variety of techniques, such as using salt in solution for charge screening. We may also control the deposition time. Generally, the depositions are run to completion. We may also change the annealing conditions, such as the annealing temperature or the annealing time. This annealing step will decrease the roughness of the deposited film and will increase the crystallinity of the superlattice. We also have several process parameters without atomic analogs. We may alter the hydrodynamic radius of the depositing nanoparticles, which affects the mobility of the particle and the lattice parameter of the deposited superlattice. We may also alter the particle hardness, or the fraction of gold in the particle to the fraction of DNA. This also affects the mobility of the particle and dictates whether or not the superlattice is capable of forming a crystalline assembly. If the nanoparticles are too soft or too hard, they will fail to crystallize and will remain in a disordered state. We may also alter the binding energy between nanoparticles. Having longer sticky end sequences or higher grafting density will increase the bonding energy between two complementary nanoparticles. So, what is the net result? Our system has the following control parameters: the deposition rate of nanoparticles onto the surface roughness of each layer after the deposition of nanoparticles, and the crystallinity of the deposited nanoparticle superlattice. Ideally, the deposition would be perfectly crystalline, with a complete monolayer at every cycle, as illustrated here. In reality, we have several non-idealities that need to be considered, including incomplete coverage or defects in the deposition, as illustrated here. This has been my presentation on epitaxial growth of DNA-assembled nanoparticle superlattices. Thank you for watching.